Hello, and how's everyone doing on the Galactic Family podcast today? We have a wonderful guest for you today, Dr. Max Rempel, um, Managing Director of the DNA Resonance Research Foundation in San Diego. Max's groundbreaking work explores DNA not just as a chemical blueprint, but as an electromagnetic resonator, a living antenna for living information, energy, and possibly even consciousness. With publications spanning genomics, biofields, and bold ideas like traces of alien genetic influence, Max is pushing the boundaries of science and spirit alike. His research challenges us to rethink what it means to be human and what secrets may still be encoded within our DNA. Dr. Max, welcome to the Galactic Family. How you doing?
Hello, everybody. Hey, Jay.
So Max, thank you for being with us. Let's start by, can you share your personal journey? How did you first become interested in genomics, biophysics, and eventually the notion of the DNA in us as a resonant system?
All right. So I'm from Russia, a Jew from Russia, and I graduated from the main Russian university called Moscow State. And my master's was in chemistry. I did my master's thesis on DNA synthesis. Then I worked for about 8 years in mainstream Russian molecular biology, studying DNA, and invented a few devices related to human genome analysis, human genome sequencing. Then I went to United States, worked in Virginia Commonwealth University as a postdoc in National Cancer Institute, also as a research fellow studying DNA and DNA and cancer and DNA and behavior. Then in Rochester University of Rochester, did another work on mice and the effects of also on DNA and effects of stress on cancer. So I did behavior, cancer, schizophrenia. So I have a lot of mainstream papers.
Yes.
And my awakening to the resonance and quantum genetics happened first gradually. I was always interested in paranormal and metaphysics and the fields. I always was sensitive to the fields, but somehow it went separate. It was like the mainstream was strong and the supernatural was something on the side, which is occasionally creeped in the life.
Yeah.
So I was aware and interested, but it was separate. But in 1999, 26 years ago, I was visiting after a big break my institute in Russia, and a friend, I never spoke to her directly, but I said hi. So it was a co-worker in the next room. She just gave me a book which was called The Wave Genome by Garyaev. So it's about quantum genetics. And I never was able to read the book because it's really hard to read this one. I disagree with it. So it's hard to read with something you disagree. But the main idea, I agree completely. So the main idea was that in addition to chemistry, there is a field. And the field is strong, the field is key, and the DNA expresses itself not only as proteins, but also as a field. And DNA is a producer of the biofield and also receiver of the biofield. It's a computer and antenna and the code at the same time. It's a code, antenna, and a computer doing the work of the biofield and doing both shape of the body, health, and consciousness at the same time. It's a big complex unit, big complex unity.
Yes, that's extremely interesting. So you're the managing director of DNA Resonance Research Foundation in San Diego. Can you tell us about its founding, its mission, and the key milestones?
Gosh, it sounds so profound. This is a nonprofit. I registered it in 2018, and basically, that's when I submit papers, you have to have some institution. So as a independent researcher, I need some institution. So that's how I called it. So it's my non-profit. And I found the research, and my previous company is still alive, still registered. It's called Localized Therapeutics. So from 2008, I went on my own, studying gradually doing part-time and then full-time research of DNA resonance. So with that company, I received NIH grants for small business, and the total was about $750,000 over many years. So it was not a lot, but that allowed me, actually, thanks to NIH, thanks to federal funding, allowed me to actually ground myself in that DNA resonance quantum genetics area. So I published papers with my coworkers, and I put the title DNA Resonance Research Foundation. So far, foundation received very few donations, but thank you, friends, for sending donations. It's under $1,000 donations. So I funded because I work elsewhere, and I fund the research.
Awesome. And your earlier work bridged on molecular biology and optics, later moving into the biofield, quantum aspects, and consciousness.
Yes.
What were some pivotal experiments or turning points in your career that led you toward the quantum genetics side?
Let's say the turning point was to realize that with that little funding, you can do experiments. And then I started heavily relying on computational genomics. Essentially, there is, when people are funded, and there is tons of funding, there are millions of molecular biologists, millions. Of my level, there are thousands. My level meaning number of citations, number of publications. But general technicians and low-level scientists, there are millions of molecular biologists who work with experiments in DNA, and the funding is extraordinary. It all goes towards developing new pharmaceuticals, 95%, and everything else a little bit more.
Okay.
But pharmaceutical industry is huge, and there is that desire to patent new pharmaceuticals, and now they expanded from chemicals to proteins and peptides and then to DNA, RNA, and complex forms of delivery. Anyway, so with that money, I mean, you need some money. It's not huge, but you need 1-2 technicians, and you need continuous funding to actually make progress. And to prove such an extraordinary thing as DNA resonance quantum genetics, you need lots more. You need like 5 people, reasonably funded for maybe 3-5 years, something like that.
Okay.
So I cannot do that, but everybody who is funded by federal or public funding is obligated. There is a rule that you have to the data you produce, you publish as a research paper, but also upload the genomics data, upload to public databases. So that is huge. And in cancer and lots of other diseases, there is a lot of good quality data. And I realized that's good enough for me. I can use published data to actually prove the DNA resonance idea.
Okay.
So we did experiments with that NIH funding. We did experiments for several years, but I have nothing to bluff about. Basically, the conclusion was always inconclusive. You can't. It's that wall. The reality resists the breakthroughs. You have to be smart to actually prove anything.
Oh, of course. And many listeners will know you from your academic publications, for example, DNA resonance signaling. Could you walk us through what DNA resonance means in your framework?
Yes, thank you for asking. Thank you for framework. Exactly. It really depends. So there is lots of types of resonance.
Yes.
And in my case, it is pretty much classical definition of resonance, except I don't define which type of the waves it is. It could be electromagnetic, acoustic, and other types. And other types don't have fixed names, but one of the names is scalar, subtle, ethereal waves, some sort of a field which goes beyond physical.
Okay.
And that pretty much is proven by others. So resonance in this case is that we have lots of DNA. Every cell, almost every cell, except few exceptions, has a nucleus with DNA. And DNA is a chemical, a polymer, pretty long polymer, pretty strong. If it wasn't dissolvable in water, you would be able to make a fabric out of it. It's very strong.
Mhm.
But in rain or sweat, it will dissolve on you. But you can extract from human body, theoretically, you can extract about 250 grams of DNA. So about a pound of DNA. And because when you dry it, it's fluffy, it would be a big part of DNA. So there is a lot of DNA. And each cell contains the same genomic sequence, 6 billion letters in each cell. Chromosomes, if you read the letters, it's 6 billion letters. The letters are A, G, C, T. Everybody knows that. So the idea of resonance is that DNA is like a radio, a computer with a radio, like a smartphone, which the cells talk to each other through waves, like smartphones. And smartphones talk to each other through resonance. There is lots of waves, but when you tune into a specific frequency and then you decipher it, now it's amplitude modulated and somehow else modulated. You decipher the signal, and then you get in a smartphone, you get the signal specifically for you. And same thing, the idea was that there is a certain radio in the cell which sends and receives the signals, biologically important signals, and the body talks to itself through wave signals, and these waves are called biofield. That's pretty much classical definition of the biofield, the waves. Again, acoustic, electromagnetic, and pretty much subtle, scalar, etheric waves. Yep.
Okay. And in more accessible terms, if someone thinks of DNA simply as a chemical blueprint, how do you expand that view? Like, what additional layers do you bring in? Excuse me. Is it electromagnetic, vibrational, quantum, and why would you bring them all in? Is it a layered system? Am I thinking of then?
Good question, but it's not specific enough. So,
Okay.
are you asking why do you think there is a vibrational level in addition to the chemical, or is it a more specific question?
More specific on additional layers in general, like electromagnetic, vibrational, and quantum, and why are we bringing like those kind of terms into it? Like, is that proven fact already that is what is going on?
All right, and then the theory relies on lots of proven facts and intelligent guesses, educated guesses. So to prove it, we need something stronger than that. But let's start from the facts. Experiments by Alexander Gurvich. It was a pretty decent sized lab. Started in Germany, then migrated to Russia around 1918, and worked in Russia until about the 50s, and then his students continue to work until now. So essentially they study biofield, how one biological experimental object affects another biological experimental object. Mm-hmm. And the classical model was growing onion roots, very cheap onion roots, grow very fast. Mm-hmm. They grow the onion, pull it out from the water. There is a growing root. And that's a sender. Growing cells send some sort of a signal. And then the receiver was either another root or it was a dish with yeast culture. Okay. So on some gel, you get yeast culture and you see the little bit of yeast culture and see how fast does it grow. And then they use the root and the receiver, and they put the prism, quartz prism between them, and split the rays into the spectrum. And surprisingly, the main bands of effect go through the air. They discovered in pretty harsh UV 250 nanometers, 230 nanometers. So there was a whole spectrum of action. But essentially, the growing root of onion accelerated the growth of the yeast on the cell culture, or the another root. So that was called mitogenetic radiation from mitosis. They accelerate mitosis.
Okay.
Mitogenic radiation, and the whole theory of biofield was built around Gurvich, and there were other people who did the same. So the idea of biofield, but the claim was bigger, that it's not only speed but also the shape of the organism which is defined by the biofield. And the most impressive experiments which I know were published, and this group worked from the 80s and then works until now. Alexander Borisovich Burlakov in Moscow State University Biological Department. I think he works on the fish department, maybe ichthyology or something, I forgot. But he works with the fish models. And they take a quartz cuvette, standard size, 1 centimeter by 5 centimeter by 4 centimeters, so about that size, and fill it with whatever fish water. I think whatever water it is. I think it's a fresh water for the fish, and then put their fish embryos, fertilized fish embryos, like 10 fish embryos in a cuvette, and another cuvette with fish embryos. And then they put on top of each other, separated by the layer of air, in a metal box, in a dark metal box, so there is no field from outside coming electromagnetic. And older embryos speed up the growth of repress the growth of younger embryos. Younger embryos speed up the growth of older embryos. The age is like 7 days versus 14 days. And if you put the one cuvette with embryos on a germanium mirror, basically, it's pretty. I have one. I hope to do the experience, but I didn't. So just a mirror, you can buy it on eBay, covered with germanium, so there is some effect on the light. Then it speeds up the growth of the embryos through the air. And if you put it on a retroreflector, I also bought those, hoping to do experiments. A retroreflector, basically, it's a set of 3 mirrors which turns the image upside down. So if the embryo, without the eyes, it's just the round embryo, early stage, looks at itself in a mirror which turns the image upside down, it is completely confused and grows multiple heads and multiple tails.
Whoa.
So that is a main proof I know of the field affecting the shape of the body.
Okay.
So now, to bring the DNA in the picture, you have to realize that DNA has the length, which is pretty substantial. If you stretch one chromosome, it's on average is 10 centimeters.
Okay.
Size. And then the thickness of the DNA is 2 nanometers, which is a lot smaller. It's the size of a few atoms. It's about 10 atoms wide. So the length brings the DNA into the macro scale, but the thickness brings the DNA in the quantum scale. And in quantum scale, everything is fuzzy, and there is a principle of uncertainty. Basically, you don't know where the atoms are. They are probabilistic. They can be anywhere, but there is certain probability they're in their proper place, but they can be anywhere in the universe.
Okay.
There is quantum entanglement, things which come in contact with each other, remember each other's state. So
Spooky action at a
protons
distance.
remember the spin, and then they're connected through the distance. And there is interference of waves. So wave particle duality is shifted toward the wave properties. And then if you look at the structure of DNA, that's my publications, early publications from 2015, 10 years ago. Saying, like, let's look at delocalization of charged particles. One charged thing it's not a particle. It's a particle, but it's also delocalized. So it's electron is delocalized. So there are it's well known. It's a textbook knowledge. In the DNA, electrons in the core of DNA, it's called base stack. They're delocalized and spread over the length of the DNA. So there is charged core, And the shape of that core, surprise, is defined by the sequence. If you just pay attention, you notice that the shape of that stack is not regular. It is defined by the sequence. AGCT, da, 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 da. So we basically build the shapes based on the sequence. And we hope to find, and we found, basically the idea was that we look for structures which repeat in the genome, and that they have, we were looking specifically for resonators. What structures of these delocalized electrons spreading, say, 20 bases, 20 letters, are repeated, and they are functional. And functional we judge by conservation in evolution. Basically, if they go in evolution up, and preferably they selected in evolution. So we looked for evolutionary selection of those sequences. And also, like if they are in important areas of the genome near the genes and gene promoters. So that's what we published. We found those structures. But also there are structures which are positively, electrons are negative, protons are positive. So we found delocalized proton structures. We call them proton wires. They're known in proteins, they're known elsewhere. So we found them in DNA computationally. And again, we demonstrated that longer electron structures and longer proton structures are selected in evolution. So we evolved to select to have optimal long chains of delocalized charges. And these delocalized charges, they are basis for vibrational oscillational resonance. So that was the main starting point.
So let's dig into the concept of resonance in DNA. In your 2019 work, you discussed the enrichment of so-called hiders or high-density resonators, high-density resonator sequences. What are hiders?
Oh, thank you for noticing. We moved very far away from that lately, but hiders are still, they're still true. Basically, the idea was that, without doing experiments, we somehow needed to look for those patterns in the DNA which are resonating. And they hide between the genes. And basically, different sequences might have the same shapes of electron clouds and proton wires. And we can use that to see if there is any interaction between them. So again, the main readout was enrichment in evolution. If they enriched, say, certain hider is enriched tenfold compared to random, that's a discovery. That's what we discovered. I think, there was like 4 to 16-fold enrichment of certain hider sequences, and they didn't necessarily have the same sequence. They were variable sequences, but same structure. So we say here, that's very likely a resonator because it is everywhere in the genome, creating the collective oscillation pattern. So we found some patterns which were predicted to be resonating. We didn't do any experiments. I hope to do experiments, and the experiments could be spectroscopic. You can put it in a again cuvette and look spectroscopy, and resonance is easy to measure once you know the wavelength. And then you can show that there is some biological significance if you find the wavelength. If you say an idea, synthesize lots of these resonators, hiders, specific patterns, activate them somehow, for example, by electricity or heat, and place next to, again, yeast culture or worm or plant, and see if there is specificity between activation of the resonator and the receiver, which is a biological experimental object.
Okay, and many people have asked, how does resonance differ from normal gene expression regulation, the chemical transcriptional? What unique phenomenon or evidence point to resonance signals?
That's cool. Great question.
Thank you.
Great question. So I think we should look at Sheldrake and Radin, Dean Radin, Rupert Sheldrake works. Mostly Dean Radin, I think, wrote reviews of biofield experiments. Michael Levin also studies. Michael Levin is unique because he has good funding and he continues to work on biofield in Boston Tufts University. I made a presentation on his lab in 2003. It's now 22 years ago. So he knows me. He follows my work. Sometimes he comments on when I send a new work to him. Essentially, it is the idea, again, proving the biofield. And there was no experiments, as far as I know, where you demonstrate that there is resonance between the DNA participates in biofield. It still needs to be done.
Okay.
But basically, where from I conclude that there is the biofield is super important. For me, it's Reiki, actually. Reiki healing is a healing modality, alternative medicine, and it was originated in India, traveled with Buddhism to Japan. In Japan, it was rediscovered, and in China, it's Qigong, and in Japan, it's like became also part of martial arts training there and healing training. But that was rediscovered by Mikao Usui, and it was always secret in the society. Like only few disciples secretly kept this knowledge. And but at some point, when Japan was losing the war, Mikao ordered his student Hawaii Takashi, I think her name was Takashi Hawaii, to bring it to the West and give it to the West. So right after the war, Reiki started traveling to the West, and now it's one of the major energy healing modalities in the West. And training is pretty simple. It's few hours. I think you have to get like about 20 hours of training and you're done. And practice, like spread over the time. And now there are other modalities like Jorei, healing touch, again Qigong, and in yoga you use again the same type of energies. There is Ayurveda, so there is, and traditional Chinese medicine with meridians, so they all use understanding of the biofield. So from there you realize that, especially I think is most impressive is acupuncture, which affects the body as a whole very strongly, and whatever traditional medicine, like whatever mainstream chemical medicines fails to fix, acupuncture fixes surprisingly well, because the chemical pharmaceuticals, they are super effective short term, but if you use them chronically long term, then they have lots of side effects. On the other hand, alternative methods like acupuncture and Reiki, they are great when you like repeatedly use it and you gradually shift your energies in the body. So it's all about energies. When we use the word energies, we mean special biological energies, not necessarily the electric.
It's like a bioelectrical field.
Joules and watts. It's not the same thing.
Yeah.
They're related, but not the same thing.
Yeah, that's very true, sir. It's like a bioelectrical field.
Yeah, you can call it bioelectrical, but I'm pretty sure there is. It's called a rational component, non-physical component. You have to have a very special biological sensor to sense it or send it, or you have to imitate biological sensor. And I wrote the whole chapter how to imitate it. It's accepted now for publication, but basically you have to have water, DNA in there, and active energy flow through that. Classical energy, like metabolism in it. So once you have metabolism and you have active work in there, then you can send and receive signals. I think it would be hard to do it electronically with purely. It's possible, I think, there are now quantum sensors. Just
Okay.
Google quantum sensor. They pretty much use quantum physical, quantum mechanical principles to actually sense the quantum fields. But it is not classical electromagnetic. It's very non-classical.
So you propose that DNA may act as both sender and receiver of electromagnetic or vibrational information. What are the implications for biology if this is correct in the terms of development, regeneration, and consciousness?
Development, regeneration, and consciousness.
Yes.
So essentially, electromagnetic treatments, and they called physical therapy. There was huge resistance from pharmaceutical industry against those, but lately, surprisingly, some of them were permitted by FDA, and light therapy is now permitted. There is a lot of light therapy devices on Amazon, and I'm an expert in those. I studied them a lot, and that was my funding. Part of my funding went to that. So most of them actually work. So light therapy is great. TENS is great. Transcutaneous electric stimulation. But you have to know how to apply it, where to apply it, when to apply it, and not overdo it, right? Light therapy, if you overdo light therapy, is okay, but TENS is not. Then there is bioresonance devices. Basically, it's biofeedback resonance devices. And megahertz therapy, millimeter wave therapy. Millimeter wave therapy hasn't reached the West, but it's known in Russia and China. So 7 millimeter wave therapy. It's very powerful. It has side effects, so that's why it's not in the West. But if you use it properly under talented healer, then it's powerful. So development, essentially, cancer is a development problem. So treatment of cancer, growing new organs, I think that's why chemical approaches miserably fail when you try to grow new organs. The promise of biology is to fix the body, and so far surgery works, but telling the body how to regrow things, regenerate, it's hits the dead end. But again, there are some developments where people realize you need to use electricity and light and sound and pulsations to actually create structures of sound and electricity and light to guide the body to regenerate. And there are some publications which now pop up where actually mainstream tries to in the lab to grow organs using sound, electricity, and light.
Okay.
So, and the consciousness,
Yeah.
That's a big step. So let's make this step. So essentially, Stuart Hameroff and Roger Penrose speak about microtubules to transmit thinking information, whatever it's thoughts, signals. In addition, like nobody cancels classical action potential in neurons. So actual potential travels very slowly compared to light. It's not electricity, it's not light, it is shifts of the ions. So ions they move perpendicular to the length of the neuron, so there is like a slow walk of the action potential. And it happens on the outside of the neuronal axon on the wall.
Okay.
Inside, there are microtubules, which they go all the way from the one neuron to another. They meet in a so-called synaptic junction. They don't touch each other, but they go all the way. And the idea is that this is an additional, I call it fiber optic system, which transfers signals fast and has much higher bandwidth.
Yeah.
More information can come. And synaptic junction is super cool because lots of psychoactive drugs affect how we think by regulating that specific synaptic junction. So the light comes, I suggest, the light comes to synaptic junction. There is some resonance there. That's the idea of Richard Alan Miller. Possibly not only him, but I heard it from him, that there is a resonance. The synaptic junction is actually a capacitor resonance cavity, and two neurons talk to each other through resonating in the synaptic junction. Now, how the DNA plugs in, it looks like the microtubules in the nucleus, the microtubules are outside of the nucleus DNA is inside of the cell nucleus. There is a nucleoli, a small organelle, where DNA somehow is tied to that, and microtubules are tied to that. So maybe there is a vibrational connection between the two. And also there is another possibility that if you look at microphotographs, microtubules go around the nucleus. So maybe there is another vibrational connection where they collect vibrational information from inside the nucleus and transmit, and this way they connect multiple nuclei of the neurons into one vibrational system. So that seems to be getting traction in the mainstream. I wrote about that some years ago, like 10 years ago, and now mainstream finally starts picking it up. It's not from me, it's just it's from Hamerov, I would say. But my highlight is that DNA is a key player there. It's not only microtubules, it's DNA. Hamerov didn't want to hear about that. He just said, when it comes to DNA, he's just blanks on that.
Yeah. So,
but still, the idea that microtubules are super. So basically, he said that this his main starting point, and his theory is that anesthetics, the one which makes a person unconscious, they do something with microtubules, and that was the starting point how he discovered that idea. And then there is some evidence that that microtubules are important.
Okay.
So then we come to the idea that DNA directly participates in the thought process, in individual consciousness. That DNA of multiple neurons connected vibrationally into one system and participates.
Okay.
And my latest breakthrough was understanding the mechanism, how the DNA actually does that. What is the main process, how DNA thinks?
Yeah.
And it took, again, like 7 years, gradual thinking and talking to people and presenting. But essentially, first I realized that when you talk about proton wires, in our computational model, we discovered that they are not only in the DNA. Electron wires are in the DNA insulated from surrounding water, but proton wires, they actually go into the water, and DNA is exchanging protons with the water. So there is delocalization of protons in DNA, and it goes into the water.
Mm-hmm.
So finally, until about 7 years ago, I just happily ignored water because I was under impression that DNA is so insulated, the core of DNA is so insulated, there is nothing happening outside. It's just an insulated, like the smartphone.
Yeah.
But then I had to pay attention to water. I was forced by chemical evidence. And then I paid attention to Luc Montagnier and Benveniste, two French scientists who said that Benveniste explained the importance of water structure, and Montagnier claimed that DNA imprints its structure on the water and back, actually.
Yeah.
And then I started looking at that, and I listened to lectures by Gerald Pollack on YouTube. And in discussion in one of the lectures, he mentioned, and I was ready to capture that, he mentioned that macromolecules, especially helical ones, would shift the layers of water, imprinting that helical structure on the water. And now I'm talking to exchanging emails, sending my chapters and papers to Gerald Pollack. He doesn't remember saying that. So it wasn't his main interest, but it was my main interest, because I was looking for the answer, how DNA interacts with water. And then I looked at Pollack's water structure, and as a chemist, I see that it makes no sense. In Pollack's structure, it accumulates crazy amount of charge, which is not happening. The small amount of charge is proven, but not like crazy.
Yeah.
I mean, otherwise, if it was that amount, it would shoot. How do you call this? Fireballs.
Yeah.
So I looked more, and I found that Pollack structure has a defect. Actually, in literally and the other way around. Literally and the other way around. Symbolically, it is defective. In Pollack structure, it's a perfect honeycomb, flat. So flat is surprising, because ice for the classical water ice is not flat. It's zigzaggy.
Okay.
And in the classical ice, the protons are in between the layers, so the layers are glued to each other. They cannot shift.
Okay.
In Pollack structure, the protons and oxygens are all in one layer, flat, and they can shift, but the layers are chemically impossible, they're too much charge. So then I found a paper by Lippincott, 1969. It's called Polywater, which is very similar to Pollack's easy water. And this Polywater has gaps. It's a flat layer with canyons where there is no structure. So essentially, it's a mixture of structured, like flat crystal water with lots of individual molecules floating around. And that structure is chemically possible. It doesn't accumulate a lot of charge because you break impossible bonds, and then it chemically is reasonably neutral. And another consequence of this structure is that if it grows on one end, it accumulates charge, it has to dissolve on another end. So it's a traveling island of crystal in the liquid water. And this structure is capable of shifting in discrete steps. One step would be in 6 directions, and 2 steps would be, relative to the first one, would be in, I think, 19 directions. So 2 steps of this polywater can encode by shifting 19 letters, 19 position, 19 orientations, I would say.
Yeah.
So that is super cool. And then I just put this DNA vertically, polywater horizontally, and by size it looks like one step of DNA equals 2 steps of polywater, and the DNA sequence would shift polywater horizontally, and it would be like click, 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 shift. So this way it can imprint its sequence on the water structure.
Okay.
So that actually the idea how to explain the idea of Montagnier, that DNA imprints in water and its sequence on the water structure. And then from that, I conclude that if there are 2 similar identical sequences of DNA near each other, they would form beautiful structure because they will be bound by shifted polywater layers. They would kind of form a very nice coherent structure, which is more stable. Okay, and I found some evidence in the computational genomics for that. Basically, those sequences are preferably selected in evolution. And then I went into the experimental data, published experimental data on how chromosomes fold, how DNA folds. And it actually captures, it freezes the positions of folded DNA. And it's well studied on like DNA double helix is well studied through crystallography, but high level folding is very poorly studied. It was never the focus of the science. Very few scientists actually studied that. So my idea was, and I have a collaborator, a friend, who said that similar sequences attract to each other through quantum physical properties. So, and my favorite resonators have to be similar, because you have to have lots of similar resonators in the cell.
Yes.
So if you talk about identical sequences, it has to be a repetitive sequence. It has to have multiple copies. And that was my breakthrough 26 years ago. I realized that if you need a resonator in the cell, it has to be in lots of copies, like 1,000,000 copies. And if you look at multiple copies segments in DNA fragment sequences, it's the most popular in our genome is called ALU, A-L-U. And it has 1.1 million copies. It's 300 letters long and 1,000,000 copies per genome. It comprises 11% of our genome.
Wow.
So that was, the hypothesis back then was 26 years ago, that must be the main resonator. That's what makes us resonate, and it's unique to primates, and humans have more of that. So that's our main resonating sequence, ALU. And I went to the conference on ALUs, met main researchers on that. And now there is experimental data. Let's test if this is true, that ALUs stick to each other. And surprise, they do. So finally, a few months ago, I discovered in experimental data of others that DNA folds by ALUs sticking to each other. So the model is very simple. DNA thinks by refolding. It's a temporary structure. It constantly goes. There is a million of them. They can stick to each other in many ways.
Okay.
But they stick to each other in head-to-head orientation. And because of inner delocalized electrons and protons, it creates new electronic structures which actually have different resonating properties every time it refolds.
Okay.
So there is a dynamic process of continuous reshaping of the water and continuous reshaping of the DNA in the nucleus. It's a very heavy solution. It's 1% DNA, 20% of proteins, very heavy gel, and it's like octopus. It continuously folds and refolds. I think it is how DNA thinks, how the cell thinks.
Okay.
It creates the field by changing the shape, and then it creates some sort of acoustic, electromagnetic, and etheric waves by vibrating with and resonating in the surrounding biological tissue.
So let's shift now into one of the more provocative aspects of your work. You've spoken of preliminary evidence of traces of alien genetic manipulation in humans. Could you overview what you mean by that phrase? Because, I mean, I have heard of a theory that the chromosome 2 was divided or manipulated in some way earlier on in our history, but I'm not sure if that is exactly what you're talking about.
It is along the same lines.
Okay.
So in parallel to my mainstream science, I was always interested in paranormal, metaphysical, mystical, esoteric. I read the books on that, and I remember working. I was 16 years old, 1980. I walked down the street near my home in Belyaevo, Moscow. I looked at the stars. The stars were pretty, and I found Pleiades, and it was my conscious choice. I will be from Pleiades. It was like, not that I discovered it. It would be like, somehow I was also writing poetry, and I said, I will be from Pleiades. It was very conscious choice decision. I will be from Pleiades. All right. And skipping forward, I never thought that aliens would look humanoid, because from my science fiction reading, they would look like anything like humans, anything not like humans. It would be octopuses, clouds, smart planets, anything, but not human looking, because there would be a variety of conditions. So
Yeah.
I didn't take seriously anything about humanoid aliens until reading the book by Lynn McTaggart. Lynn McTaggart. The book is called The Field, and I read it in 2009. I think it was published around that time, just before that. And it talks about quantum nature of reality, that nothing is real, everything is um, follow on macroscopic level follows the quantum principles, principles of uncertainty, observer effect, delocalization, interference, wave particle duality, and so on. So there is a lot of smart people talking about that. She interviewed lots of physicists, and that was super great. And then Dean Ryden was there, and Sheldrake was there. So I was initiated. It was on my birthday when I closed the book. There was huge gap. If everything is so non-solid, so wave-like, let's check out the aliens. That was that simple.
Right.
And I dived into YouTube. 2009 YouTube was already pretty big, and I started looking at pictures of aliens, and I recognized them. Actually, I recognized the grays. And and then I googled ufology, Rochester, upstate New York, and I found next few days there was a meeting on Wednesdays. Richard Dolan and Cookie Stringfellow were guiding the meeting, and there was pretty big group in the Barnes and Noble bookstore, Barnes and Noble. And as usual, I was like 5 minutes late to the meeting, so I sneaked, and somebody was already presenting. And it is my friend now, and I took many hours of interviews, maybe 15 hours of interviews, something like that. 8 to 15, many hours of interviews of her. She is the best abductee experiencer I know. She remembers everything. When they were starting, they asked her, do you agree to participate in saving a race? And she still till today doesn't know if it was saving a gray race or human race. She doesn't know. At that time, she thought it was about, I don't know, either one of those. And so, but she said, I would agree if you let me remember stuff. She was smart enough as a child. So she remembers stuff. And her experience is extraordinary. She remembers a lot. So I took interviews. And then I found Bashar, which is an excellent channeler. And if any in the audience, you don't know what channeler is, it's like medium or telepath.
Yes. Great channel.
But you telepathically communicate remotely with some aliens, dead people, discarnate people, spirits and deities, and God, and angels. And I don't know, I wouldn't recommend talking to demons, but once we got a demon also. So I was looking for channelers, and it was interesting. The first channeler spoke to me. He said, we are Pleiadians, you're one of us.
Huh.
And I didn't believe that, because that was too trivial. I thought it was a trick. Now I know it was true, but I didn't believe that.
Okay.
All right. Second channeler I spoke to, it was very short conversations, but second channeler, I said, I have so great ideas in genomics and DNA resonance. Why nothing goes right? Experiments fail. I don't get funding. Nobody helps. And I jump from one idea to another, from one experience to another, never actually doing anything of substance. It was many years ago. It was 2000, maybe 11 or something, 10 maybe. And the being from the other side said, later you will be designing a species. You'll be designing a new species. And I thought, oh shit, I want here in this life to achieve something. I don't want just to learn it here and maybe some other life to design another species. So another race. So I take it seriously. Those were the first messages. And then in 2013, I got my own channeler. For 10 years, we spoke at least once a week, but mostly twice a week. We spoke, everything is on record. Pretty much everything is published. We usually publish like right away. Most of that is on video. Some of that was on sound only. James Charles, Human Colony, Jim Charles, Jim Charles, Human Colony.
Mhm.
Check it out. And so I had lots of opportunities to talk to aliens. I understand the limitations of channeling. It only works on need-to-know basis. If you need to
Mhm.
know that, you get it. If you don't need to know, it's like complete nonsense. But if you ask the right questions, if you change yourself so you actually need to know, then you get it.
Okay.
So we spoke to, and this channels lots of entities, hundreds of entities and spirits. So we spoke to everyone, including my favorite John Lennon and JFK and Marilyn Monroe and lots of Russian people and Francis Crick, the discoverer of double helix, and Gurvich and lots of others. So we had lots of and deities as well. So the long story short, I read a lot of books. I spoke to a lot of people. I started in 2009 running my own experiencer support group. So I had my own experiencers sharing the stories in the circle. So long story short, the idea of ancient aliens is true. The aliens bred us. They took some of the species on this planet, mixed with their own genetics, and kept improving, changing. And usually when they create a new human race, they place it elsewhere and make them conquerors, so they conquer everyone around. So they make super soldiers, or they make workers to do the work for them, and they plug in the idea that the humans should treat aliens as gods. So that is implanted in our sequence, that believe that we should be faithful to aliens.
Okay.
And that we should work hard. These are a couple of major artificial properties put in humanity. One more. Be a good warrior, follow the advice from the aliens, treat them as gods, and work hard.
Okay.
So, and then we know from lots of sources that in the 40s and 50s, there was agreement between English-speaking countries, America, England, UK, Australia, New Zealand, and the aliens, mostly Greys and some reptilians, some Orions, and some others, that in exchange they give us technologies related to the military, and in exchange they get the right to hybridize in secret. And first they started hybridizing the enlisted, active duty people in the army, air, and so on. So they were like ordered to keep silent and not to mention that. But then they expanded it to general population. And currently, I estimate it's maybe between 2 and 10% of population are recent alien hybrids. There are ancient alien. We are all ancient alien hybrids, and there is influx of recent alien hybrids. So the secret alien hybridization, alien abduction happens. They take both parents, maybe separately, maybe together, take reproductive cells, eggs, sperm, manipulate, maybe grow a little bit in the outside, then take the mother again, impregnate the mother, put her down. Then sometimes they again take out the embryo, grow again in the spaceship, and then it becomes grown in alien culture. So this would be outer, external, extraterrestrial hybrids, which are hybrids between us and aliens growing up there. And there are local hybrids which are born. So basically they're impregnated. The mothers are impregnated. The hybrids are born here, and lots of them are born as normal humans, and they discover that they are alien hybrids either later or never, or some of them are born with the knowledge. So there is a lot of variance.
Okay.
And and then we come to the very simple idea. So the idea came from question. The question was asked, how do we prove the aliens? How do we prove extraterrestrial visitations? The question was asked by Ross Coulthard in summer 2024, a year and a little bit more ago.
Mm-hmm.
He announced it on YouTube, and he said there is some funding. And when we hear the scientists hear the word funding, then our brain starts working really well. So I thought, yeah. And my friend from Australia, Swami Dave Little, he introduced me to Coulthard. So actually, I submitted my application right to Coulthard. It had lots of ideas, and one of them was if the abduction and hybridization happened recently, it would just take two parents and an adult child, look at their sequences, we should see the traces of manipulation.
That's what I was about to ask too, was what data sets or genomic signatures are you analyzing to search for that possible alien or non-parental genetic contributions?
Right. So, yeah, I used the number of sources was limited, and now it grows. Let me first pronounce what I really need. We need both parents, absolutely necessary, and at least one adult child.
Okay.
It's called full family trio. Without parents at the moment, I don't have enough information to tell if it's alien DNA or not.
Okay.
Because from what I know from the aliens and from whatever abductees tell, this is the same DNA. We are being visited by human-looking aliens, which many of them are closely related to us, and all humanoids which are visiting us have the same double helix with the same letters. We know, again, from channel sources and personal communications of experiencers, that there are life forms which are not humanoid or which are not based on DNA. It's possible, but we're being visited by humanoid aliens related to us. That's why the hybridization basically modifies the sequences, and some of them are just modifications of our sequences.
Okay.
So we are looking for insertions, substitutions of parental sequences with non-parental. In classical Earth scenario, the child always has two chromosomes. There's a pair of chromosomes called homological pair, and they are one from mother, one from father. It's always 50-50.
Mm-hmm.
And nothing else. It's not known to biology that we should carry anything else. And so we just check the sequences of the parents, the sequence of the child, and see if there is anything inserted or substituted. Mm. And it's very trivial. There is no complicated math. It just volumes are pretty big. So if it is genotyping, basically only looking at known human variants, it's usually a little bit under 1,700,000. It is a decent sized file. It may be less than a megabyte file. Okay. It's something you can easily send by email through Gmail. So genotypes, and then genotyping is just looking at known variants, and sequencing is actually you do the whole genome sequence and looking between known variants. And so far I didn't look at any sequencing, but I have one experiencer abductee family, self-report. They all self-reported. Remember, self-reported.
Yeah.
I don't have a proof, but we could do hypnotic regression, and then it would be some additional proof that it's actual abduction.
Okay.
So, one family, they pay for themselves. They're very interested. They plan to sequence all members of the family, 2 parents and at least 1 adult child, and let me look at the sequences, and then we will look between human variants, which is much more interesting. So initially, there is in open access, I found only one data set, which is called 1,000 Genomes. It's called 1,000 Genomes Project. It's a very old one. And I was looking for insertions, and I found insertions in 2% of population. I was very excited. I immediately wrote a report, and at that moment, I couldn't find any flaws in that. It looked to me like pure alien insertions in 2% of populations. They were super crazy, super unusual. It was unthinkable that they could be produced by anything else, because it was substitution in both chromosomes at the same time. Normally, it would do, if it was a random mutation, it would be in 1 chromosome, but in 2 chromosomes, the same substitution was too unusual.
Okay.
And I self-published, just uploaded to public self-publication sites like ResearchGate and Vixra, and I sent it to that place, which the funding source in Australia, and to Ross Coulthard. I haven't heard back from them. And then I started digging later into that, looking at more data on that database, and I realized that there might be some technical artifact. I still don't know if it's alien insertions or technical artifacts, but it could be technical artifacts.
Okay.
Because this data set is old and was done using old methods. Could be some weird coincidence. I cannot say that it is real. And my publication clearly says preliminary. So preliminary. At that time, I didn't know, but now I see possible artifacts. Now there are much better data sets, but they all require application from an academic institution, and they review your application for a long time. So that would be one possibility. Another possibility is actually much more interesting, is to genotype abductee families. So that publication was published May 30th this year.
Yes.
Self-published. And then in October, I think 6 or so, almost half a year later, it was picked up by Daily Mail. I got like a random email. I get lots of random emails, and it didn't even say my name. It says I'm blah 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 from Daily Mail. I didn't believe it's real. I thought it was spam. So I said, "Are you a spammer? You didn't even give me your name." No, it was a real person, and I still wasn't sure it was real. But he didn't give me enough information to prove that he was real. But anyway, I answered to his questions by email. I was busy with other stuff. I was discovering the other stuff, actually, that ALU pairing. So I didn't pay much attention, but I answered questions by email. And he said he's in a hurry to submit it to his stuff. I didn't even know what Daily Mail was, so I wasn't thinking it was important. Anyway, it came out, and I started getting. It came out maybe 3 weeks later or something. I started getting emails from people, and again, they were strange, but even stranger was they were referring to something I don't understand.
Yeah.
Finally, a friend told me, like, you were mentioned in Daily Mail, and I Googled what Daily Mail is, and that was made rounds. The rounds actually faded out. After a couple of weeks, everything faded out. So there is total maybe 10, 15 mentions of that. And I was invited to a couple of interviews already, two or three. And basically, I tell them what I tell now, except it was much shorter. It was mostly the last part that I am not sure if it is real. It could be either way. We need to analyze more data, and at the moment, I don't have more data. I got actually one more data set. I analyzed it. So one more family, abductee family. So I have now 3 abductee families. All of them are adults who gave me the data and permitted to anonymously publish.
Yeah.
So that's what I'm looking at, but I'm looking very closely because there is so much interest. So is this I got 15, now maybe 17 letters from abductees who want to be studied. And I looked at the prices. The prices are really good for genotyping. It's $50 per person. So
Okay.
father, mother, child, adult child would be $150. So for 2000, we can analyze about 13 families. It's that cheap,
Yeah.
and that's already something.
Oh,
And the
yeah. No, that'll
hypothesis,
give you the evidence you need right there.
and the hypothesis is that they would share some modifications. So first three families, there is zero shared insertions between the families.
Okay.
Each family has its own set. And the sets are very interesting because some of them can be random, but some of them are, like in the best case scenario, it was, I think, 9 insertions sitting in a tight area on one chromosome, which chromosome 4 at the end of chromosome 4, like spread over. So that is unlikely to be an artifact. Possible, but unlikely.
Okay.
So first thing we need to do is just to regenotype the same family. They all submitted the informed consent, so we just ask them to send again. So the procedure is very simple. They order, or I order the kits. The kits arrive. It's a brush and a tube with a ethanol detergent. You brush inside the chick, put your brush in the thing, wash it thoroughly, trash the brush, close the cup, put in a box, pre-labeled, send it out. I wait, I register, take a picture, send it to me, so there is a code. I register that code on MyHeritage service. I download the data, I can share the data with them, analyze the data, look at insertions, what is in the child which is missing in the parents, and that's it, and anonymize and publish the collective result. So that's simple. So total for genotyping, it's about a month or two. For sequencing, it's the same procedure. For short read sequencing, we can look at substitutions, but with a cheap sequencing, it's called short read sequencing. It's $500 per person, 10 times more expensive, $1,500 so per 3 people, and then the analysis is a little longer. It's maybe 2-3 months. There are commercial labs that send you the kit. You do the same procedure, you collect from the inside the chick. And there is long read sequencing. Pretty much it looks at the insertions, and it could be non-human insertions, essentially. It's 99.5% of the genome, very thoroughly sequenced with long reads. It's better, actually. I have lots of knowledge because I was a director while doing my independent research, I was a director working full-time as a director of sequencing facility. We did long read sequencing. So through my hands passed lots of the samples and customers, hundreds. So I know the technology really well. So with long read sequencing, you can look at insertions into human genomic and reliably place them. The problem why short read sequencing is not good enough because you use human genome as a reference, and the short reads, you don't know if it's insertion, you don't know where to put it. It looks like errors, but if you long read, you know where to put it because insertion is surrounded by known sequences. You just take the sequence and put it in between. So long read costs $2,000 per person, so total $6,000 you can do the family. So we are raising money, and families can fund their own analysis, and I will give you the results. And I got a partner, Natalie Hill from Tennessee. You interviewed her.
Yes.
And she's a founder of United Experiencers Initiative.
Yes.
And she knows a lot of people in ufology, a lot of abductees, a lot of experiencers. So now we need basically to raise funds. The money don't even need to go through my hands. It can go through Natalie or directly to the families. They can order the kits themselves. So we, and go ahead. Do you want to ask a question? I can jump to the next topic.
I'm waiting. I'll just wait for you to stop, sir. You could talk forever on this because you know a lot about it.
Let me say one more thing. And you want to make a movie. You want to make a documentary, a non-fiction documentary, where we would interview abductees, collect their stories, do hypnotic regression, videotape the hypnotic regression, what do they say when they're under hypnosis, and analyze their DNA and actually tell what we find.
Oh, wow. So, in your view, would a hybrid human alien genome show different resonant properties like the vibrations, electromagnetic signatures compared to the normal human DNA?
Wow, that's a good idea. I didn't look at that. I have candidate sequences, which my favorite candidate for 26 years is that ALU sequence, ALU. It's called transposable element, transposon. We have over 1 million copies. It's 10% of the genome. So if your genome is 250 grams, it's 25 grams is ALU. And that's my main candidate for resonator. So I expect that aliens, when they mutate humans, they would place their variants right in the ALUs, because that would change how genome folds and how it resonates.
Okay,
and to do that, we need long read sequencing. Genotypes look between the repetitive sequences, and short read sequences possibly could see some of the modifications in ALUs, but mostly they go outside. Like all short read sequences goes between the repeats, and repeats are 55% of the genome is repetitive, so 50% is repetitive. So short reads usually look between them. So we need a little more funding to look at the actual resonance sequences, but still there is lots of information, even from genotype and from cheap one, we get lots of information. And I think that practically we should genotype everyone, look at most striking unusual insertions or substitutions, and then we sequence them short reads, and then we sequence them long reads.
Okay.
Go from $50 to $500 to $2,000 per person.
And if resonance is built into DNA, might hybrid or alien DNA also act as an antenna for different information fields like consciousness or interstellar information? Is that where we might be getting more information, the genomes are acting as a transceiver receiver for something else out there, to like the spooky effect?
Exactly. You hit another point which I should have mentioned. Thank you for bringing it up.
Yes.
I believe that hybrids are different, starseeds are different. There are savants, and the main abilities that are brought by aliens is telepathy.
Okay.
Telepathy. And this brings up the telepathy tapes by Kai Dickens, the telepathy tapes by Kai Dickens. And the main discoverer of telepathy is Diane Hennessy, the main scientist behind the project. They basically claim, they already demonstrated a lot. They are exceptional. It's super interesting to listen. But they also claim that recent experiments, which were funded through making the movie documentary, allowed them to do properly controlled, double-blind experiments in separate rooms, where the sender and receiver are in separate rooms, and the signal is generated computationally and captured on video how the signal is absorbed by the sender, transmitted by the receiver, and the receiver actually types it up on the keyboard independently. So that is a definite, as it should be, definite proof of telepathy in autistic, nonverbal, non-speaking autists. So, and my claim obviously is that most of these autists are likely hybrids.
Okay.
So that's kind of obvious. When I heard about that first time, that's what I wrote. Kai Dickens, Diane Hennessy, these should be alien hybrids. And they didn't answer anything positive or negative, but I think they said at the moment to themselves that, gosh, it's just too much.
Yeah.
Proving telepathy is one thing, but saying that we are visited by the aliens and being hybridized to bring telepathy into us is is just another level of strangeness of being on the fringe. So they, but Diane Hennessy was there. I was there in the audience at Contact in the Desert recently, and I was in her presentation. So one of the main claims, and it's like, wow, by Kai Dickens and telepathy tapes, is that these telepaths, autistic telepaths, they are connected into a network, telepathic network called the Hill.
Yes. So
my estimate, there are hundreds of thousands of telepaths in that network, and most likely these are hybrids.
Okay.
And we have been genetically modified to evolve in a new species.
Okay.
That was claimed by Bashar for the last over 25 years, almost 30 years, as he speaks, that we are birthing a new species. He named it Homo galacticus.
Oh, wow.
The humans that went into the galaxy.
Okay.
The hybrid species. And
so is that like the hidden code within us then? Like, is it simply resonance, or do you believe there's embedded information like universal in our genome? Because I know DNA can hold information as well too.
Yes. So the politics of that, I think, is a little more complex. You have to really understand why do they do that. I think we are overdue to developing telepathy, and even if aliens wouldn't force the telepathy into us, we would be pretty telepathic, but we are repressed by our culture. Our culture is especially repressing telepathy through chemistry, electromagnetic waves, cultural upbringing, childhood experiences, childhood trauma, money system, lots of other things. Like research into telepathy shows that tribes which are outside of the Western culture, they're telepathic. Like hunters go into the trip and exchange the news with a base camp. Yeah. And if they need it back, they hear that telepathically, they come back. So Amazonian tribe, Australian tribes, they're telepathic. African tribes, the ones which are still wild, they're still telepathic. I also know that humans have been taken many times, Homo sapiens have been taken many times to live out there, sometimes as individuals, sometimes as whole populations. And there is a population in Pleiades which are pure humans from 2,000 years ago, and they were telepathic. When they are in the telepathic culture, we are naturally capable of telepathy. It's only the culture, chemistry, ecology, and stuff which is repressing the telepathy in normal us, in normal humans. So the reason the aliens modify us, they want us to become telepathic, and then we become much more interesting for them to incarnate here. So they create a civilization that would be fun to incarnate, in which it would be fun to incarnate. Pretty much all of them, they want to be incarnating here, and they want us to be more like them. Reptilians want us to be more reptilian, Pleiadians more Pleiadian, Greys more Grey. Yeah. So they try to, they're like multiple races, everyone creating hybrids to push us towards their race. Okay, so and the main achievement of this hybridization program is basically, in autism, they block speech. If you are not speaking, you have to be telepathic. So that's pretty rough, pretty harsh, but that finally created a population, small population of people, maybe a fraction of a percent, maybe 1/3 of a percent, which are telepathic, which create a telepathic network around us. And another news from the telepathy tapes is that they teach normal people, which are called neurotypical, they teach neurotypical people to become telepathic. So their teachers sometimes answering the questions without listening, without hearing them only in the head. They receive the
Yeah.
questions in telepathically and then answer in the voice. So sometimes they do the lesson in silence. So that already is happening. And good news is that nice people who are less selfish, more altruistic, and more kind are much better for telepathy. Essentially,
Huh.
selfishness and egotism is blocking telepathy in us. You have to be relaxed and altruistic and meditative to be in telepathic state.
Yeah, we have a lot of interesting subscribers, that are listening right now. We have, Lady Daniel. She is she believes, and I full-heartedly believe her too, that she is a hybrid with hybrid like DNA structure, and she should get a hold of you and do that test with the mother, father, and her DNA, and I believe it would help her to know what was hidden, that in her DNA could be the key to understanding everything.
Yes.
And we also have a lot of people that have had telepathy also with different people in this group as well. So you're speaking their language. Now, how do you see the interplay between consciousness research, biofield research, and genomic research and hybrid genetics in the coming decades? Is it going to be more focused on the genetics and resonance focus?
Yeah, I think Bashar, I trust Bashar. Bashar so far was very precise. His estimate is next couple of years we'll get open contact.
Oh, wow.
At maximum, our current status is that 2 to 5 years we'll have open contact. And then we gradually become integrated in Galactic Society, and maybe in 25 years we become a member of Galactic Society.
Okay.
So my understanding is that prediction of the birth of the new species was not only Bashar, it was Blavatsky as well. Helena Blavatsky, 1882, was prediction that there will be a sixth human race called, she didn't give it a name, but there would be a sixth human race coming. We are number 5. And I think that race will be us hybridizing with aliens, not only through abductions, but naturally. I think they will start coming down. And actually, I think we already have, according to David Jacobs, we have thousands, maybe tens and hundreds of thousands of hybrids working among us who were brought up there and came down here to live. And they are secret carriers of alien culture. So maybe they will come out. Maybe we'll be surprised to find out that we already know some of the hybrids, and maybe some of the hybrids we didn't know, but they will be among us.
Okay. So do you have a story? Could you share a story, a case study or data set where something looked off in the genome under your model and suggested an anomaly worth further investigation?
So far, my analysis is super primitive. I'm just look how many non-parental insertions are there, and there are tens of them.
Yes.
But I'm looking only in like talking about 3 families, just 3 families, only genotypes. So we're looking at only known human variants, about 700,000 human variants. And we look at non-Mendelization, basically non-proper inheritance. So if both parents are AA, and the child is AG, so this G popped up from nowhere. So that could be a trace of alien manipulation.
Okay.
Maybe they took a piece of hybrid DNA and substituted the piece with human, so we see that trace of manipulation. And if there are, as I said, 8 or 9 markers at the end of chromosome 4 in one of the families, they're like in tight region. So we might look at that region and see maybe there is already known function there. But again, my understanding is that looking at protein coding genes is not that interesting, because proteins is not everything. Consciousness is not written in proteins. Consciousness is written in ALU elements between the genes. So we need to look at intergenic sequence. There is very little I can tell about intergenic sequence at the moment. Once we get full genome sequencing with long reads, that would be much more telling. And if we tell, like, what we can do, like this group of abductees, they know from their hypnotic regressions that they are Pleiadian hybrids. These know they are Arcturian hybrids. These know they are gray hybrids. And we can see what is association of this sequence variants between what they know and what is in the sequence. It still needs to be done. I have only 3 families. For such an analysis, we need maybe 3 groups of 30 families. That would be at least a good start. So we need to grow that. So I'm inviting.
That's the whole work of science. I mean, that's what you're doing. You grow it from the bottom up, and you've had 1, then 2, then 3, then hopefully you'll have hundreds. Yes,
and I would be happy to share. The computational analysis is very easy. I share the programs. It's the program's already uploaded, but you can talk to artificial intelligence, and Claude is very good in programming. So Claude paid version for $20 a month. You can program yourself and double check by yourself. So I would be happy if other researchers would do the same. And actually, abductee families, you don't have to send your DNA anywhere. You can do it at home and analyze it. So I think the competition now will happen between different races for the right to send the hybrids down below. And that humans will have a choice which races we will invite, and how much we want to hybridize. And at the same time, there is an agenda by secret rulers to introduce AI and merge AI with a human and give every human instructions and guidance how to live. So there is that competition between more authoritarian and hierarchical society and more open society. So awakening to the hybrid program and the histories of different races would allow humanity to be more educated in the fact, in the choice which hybrid races we allow to land and to hybridize with us. So the competition.
So what are the implications if your hybrid alien genetic work is validated for humanity? Is it going to change our identity, medicine, consciousness? Is it going to be disclosure maybe?
I hope to help disclosure. And so far, the hybrids is a minority. And but there is a hybrid culture, starseed culture. We meet at festivals and conferences. We meet at yoga studios. So there is a culture growing up. And I think we are not the future race. I think we are intermediate between the future race. The future race will be that hybridized race where humanity will allow the aliens to actually land and marry humans, mate with humans. So lots of humans would be able to actually travel abroad, travel to the galaxy, and that will be basically open society with a lot of hybridization happening naturally. And one of the messages which is I think is super important is that there is a certain amount of hybridization which is healthy for the biofield of the planet, but then excessive hybridization would be catastrophic. So that we learn from the history of the Greys. They destroy their genome by excessive genetic manipulation. And genetic manipulation on this planet already happens for the animals and plants. And I'm pretty sure that it happens secretly for the humans. Lots of totalitarian rulers, they clone and hybridize, making super soldiers.
Yes.
And also they need telepathic spies. So this hybridization program was done by the military of the planet to develop telepathic psychic spies and telepaths to interact with the aliens, and actually psychic workers who would invite the craft and then allow to reproduce the technology.
Okay, so for our Galactic Family audience, suppose you were to speak directly to any extraterrestrial intelligence listening in. What message would the DNA resonant side of humanity, what would we send? I know I hit you.
There is nothing special, actually. I understand that they have all of them pretty much are telepathic. All of them are very advanced, and basically, I think it would be great if they educated humanity on the dangers of excessive genetic manipulation. And I wrote the whole book for them, actually. The book is called "Welcome to the Earth: A Guide for Aliens," and it was written in 2013-14. After I spoke to them through channel, and I realized how little they know about us, so I wrote the whole book where I proposed the plan, and I think they already know the plan. They already know the book, so my message is already pretty much articulated. But since then, what came up is more genetic manipulation and more AI manipulation. So the danger is basically everybody tells us. The Greys tell their story of the genetic basically destruction. They destroyed their biofield and made their genetics very incapable of reproduction. So everybody knows the story of the Greys, and that lesson should be learned, and we should avoid excessive use of AI and excessive genetic manipulation. That should be, we should become literate in collective vibrational biofield.
Yes,
that secret should come out. Did I answer a question? I guess that's the main message.
Yeah, definitely, sir. I'm sorry, Dr. Max. Definitely. I'd see I told you, it's ingrained. It's hard to get out of here. But so what unanswered questions or dark corners of your field are you most excited about regarding the hypergenetics and hidden codes?
Right. So naturally, science-minded people, we think a lot, and that thinking usually is attracted by dangers. So the danger of the collapse of the collective biofield and us becoming the Greys is an attractor.
Okay.
So, and when you start thinking about secret rulers and the secret agenda of the Greys to convert us to become managed by AI and become cyborgs, that is so painful, so scary that it blocks us. So I learned to live with that in the last many years, like whatever, since 2009. I learned to live with that and stay positive.
Okay.
And I guess the main understanding is from psychedelic culture, from psychic culture, from metaphysics, is that it's all a game. It's all an artificial reality. It's not computational artificial reality. It's natural artificial reality. It's natural system where there is a lot of players, and we are souls which incarnate here to play. So we are actually in very good hands. There are angelic beings, deities who manage the whole reality. And this artificial reality is geared up, it's optimized to have as much drama as possible. So
Yeah.
drama will be always here, will always be here. And it's like collective drama and individual drama. But yet they like the game. The Earth is a wonderful playground. It's a wonderful server with a wonderful playground. So we are due to an upgrade, but they don't want to crash the server. They want to upgrade it as we are here and to keep the drama to maximum. So
Okay.
so take it easy. We're in good hands. We are trusting the aliens and angelics and the higher beings that run that simulation. For example, like there is a New Jersey drones which came out around over a year ago, and they are hanging around nuclear facilities, demonstrating to the secret rulers of the planet that the global atomic war is not permitted. The global atomic war was scheduled, obviously was scheduled at the time the drones appeared over a year ago, and it's not permitted. So plan A is not working.
Whew.
So plan B maybe is coming, but go ahead.
So is it going to be like a false flag event, like everybody's saying?
It was. I think the atomic war was planned. False flag event is obviously is planned. So
Yeah.
so we'll have more surprises. False flag attack, another infection, AI taking control and doing something nasty. I think we should see. Let me back up a little bit.
Yeah.
The free will of humanity is respected. Otherwise, the game wouldn't be interesting. And if humanity didn't have free will, if our individual and collective choices weren't respected, it wouldn't be such an interesting game. So we are given a lot of illusion of free will. We are being secretly manipulated by all different forces, positive, negative,
Okay.
all sorts of colors. But we should choose to give AI certain place in our culture. And for that, for the collective to choose that, it has to see the dangers. It doesn't see the dangers yet. So there will be probably overuse of AI in a certain layer of society. We will see how it finally screws up, and then we will back up to actually get it right. I know in Pleiadian culture, the UFOs, the ships are conscious. They are AI, and they're given their own right to think and to live. Their home instruments like robots are conscious.
Now, do they
And
have
at
to
the
be a
same time, they
robot, or can they be an organical AI system?
yeah, many of them are organic.
Okay.
Yeah, many of them are biologic. So you give them life and give them respect.
Okay.
But Pleiadians from Era, they live pretty biologically ecological life. So that's my aspirations are that we are going, at least a part of our culture should go in that direction. Also, I understand that there are multiple timelines,
hmm
and each of us is attracted to a proper timeline by our own vibrations. So keeping the body and the spirit and the mind in a proper vibration is super cool. And this way, you just jump timelines and go in the timeline you like. So the fact that we ended up, you and I and audience, we ended up here is because we are of that vibration. And once in a while in my life, I noticed, oops, this morning I shifted the timeline again. It's slight shift, but I not, and sometimes it's during the days, like click,
yeah.
like boo. Like I don't know if you notice that, but once in a while, between in a period where nothing is interesting happening, there is that click, and you hear that the reality is slightly
It's
shifted, that refreshed.
I have heard. Okay, this is what I've heard. Okay, I'll be walking, and then all of a sudden, like my right ear will go completely silent, and I'll hear like this buzzing noise in it,
Uh-huh.
like a ringing or a humming or something, like a bzzz, or something, and then all of a sudden, my hearing comes back like a few minutes later, and I'm like, what was that?
Uh-huh.
Is that what you're trying to?
Yeah. Do you have like a trio of two parents and adult child to the genotype? Let's genotype you.
We have two, mother, father, two children, both both male. Me, my brother's younger, and I was born in 1985.
Why don't we genotype your family? Can you find some funds into that?
Yeah,
Just ask your audience to so your parents are alive, and I need informed consent of all participants. Would your parents consent to be part of the study? I wouldn't publish the names, but that would be super cool.
Yeah, they
First
definitely
we do
would.
like cheap genotyping, but then maybe audience will fund the sequencing. So that would be super cool.
Yeah, I would love to do it. And then also, when I was 16, I actually had 2 hours of missing time. That's why I asked you about the living electronical system, because I believe that I was abducted by a living spaceship.
Sounds about right. Lots of them are living. Yeah, lots of them are conscious.
Okay, wow. So on a final note here, what would you like to see accomplished in the next 5 to 10 years in your research, quantum genetics, hybrid DNA, etc.? And how can the broader consciousness explorer community support you in it?
Okay, so the plan with extraterrestrial genetics is pretty straightforward. We need more abductees, more experiencers to genotype, and then hopefully we'll get hold of alien samples, maybe from elongated skulls and other mummies and other archaeological discoveries, and maybe some of the hybrids could donate their DNA. And if hybrids are conscious of their hybrid origin, that would help us to actually trace. And then finally, the genes probably will point to telepathy genes and psychic abilities. So this would be in between the protein coding genes. It would be non-coding sequences, most likely inside the allos or inside the other repetitive elements would be some modifications that would allow the aliens to activate psychic abilities in humans. I think that will go in parallel with humanity awakening to telepathic abilities. So that population of telepaths, starseeds, and psychics will eventually become more organized, more powerful, and it doesn't have to have the money. It could be some other layer of society which just doesn't care about the money, like autists, nonverbal autists. They don't care about the money. They just are living their own lives. So there will be some development when we discover there is lots of hybrids around us. There will be new relationships between so-called ancient hybrids and modern hybrids. There will be gradual shift, or maybe it would be immediate shift.
Okay.
Ascension is the main topic of the messages of the aliens. So ascension is my highest aspiration, that we collectively just rise in vibration and go from survival mode to creative mode to unity mode, where we become more like other civilizations which made it from non-telepathic state to telepathic state. So maybe I think if all inhibitors like ecological, chemical, cultural, all inhibitors of telepathy are tuned down, maybe we'll wake up, like pop up, lots of telepaths will pop up around. So I practice telepathy with babies and dogs and cats and other animals. So that is super cool. Plants, ocean, dolphins. So lots of telepathy. So I think that will gradually, like telepathic society will just grow in between human society, in between traditional mainstream culture. Okay. In terms of DNA resonance, that's a resonance medicine. It already exists. It already progresses. So I would imagine like like just to give you a sense of that. So we did a channel session of alien healing. There was Arcturian, Pleiadian, reptilian, Yael, few other. It's all on tape. And every type of healing, when a channeler is possessed by an alien doing the healing, you see the technology is very different. The focus is very different. So once we are ready to accept these healing techniques, that would be just a revolution in the medicine. So giving more coherence to the body. It's all about coherent vibration in the body. So it's purification. We already do a lot of our community does a lot of sessions with psychedelic medicine. So I would imagine that that will introduce some DNA sequences as well. So
Okay.
the forms can be different. It could be the gloves of the healer might be electrically charged with the batteries and send some DNA vibrations. I would probably put some synthetic DNA or natural DNA inside the glove so you can move the energies in the body.
Okay.
Sensors could be DNA based. So it's very close to, if you Google quantum sensors, it's very close to quantum sensors, but with the specific DNA sequences, you would be tuning into specific DNA sequences, quantum sensing this vibration. And it's very close to acupuncture and traditional Chinese medicine and Ayurveda. So that would become more mainstream naturopathic medicine with the use of DNA vibrations.
Okay. And if listeners want to explore the work of the DNA Resonance Research Foundation or collaborate with you, what are the best ways to reach you, follow updates, or get involved with you, Dr. Max?
I have a ADHD and I'm substantially autistic. So my favorite form of communication is telepathic. So connect to me telepathically, and I can say no, or voice messages on all possible platform, Facebook Messenger, WhatsApp, Telegram Messenger, and Signal voice messages is easy because I'm always busy, always doing stuff. But when I'm walking the dog or driving, I can play your messages and dictate the answers. And that's how I work with my collaborators, mostly through voice messages. But, text messages, email, like as a scientist, I'm all in email. So email is great. So I'm pretty much, lots of different types of communications are great. You can find me, well, here is my xg1.org is one of my websites, maxrempel.com, xg1.org, dnaresonance.org, and hucolo.org. These are the main 4 sites I manage. And there is one Russian, which is tamza.org.
Okay. And we have...
Sorry, tamza.com. Yeah, okay.
We have some questions from the audience here. Curious as to the role of viral payloads shaping the biological organism. Does Dr. Max have any thoughts on symbiosis between humans and its environment?
Okay. I would say don't obsess with the dangers. My take is go to yoga, be with the people, help other people. And what will be, will be. And if you discover a conspiracy, surprise, it is as it is. It is a drama. Viruses are part of the drama. When we moved to Washington, D.C. area, very close to Fort Frederick, there was crazy number of infections attacking us. We came from Russia. It was just super, like one infection after another. It was like 3 years of getting used to infections. I think the experiment, they seeded right on population right there. It's, I think they experimented and they continue experimentation. So I think most of the infections walking around are intended as upgrades or changes of human person, and you can fight it and become sick and paranoid, or you can just go through infection as if you would go through a psychedelic trip. That's
Okay.
my favorite. You get fever, you enjoy it. Breathe it,
Okay.
enjoy it. It helps you be partially somewhere there, partially in the body. And I take pain as a part of the drama.
Okay.
So if you are playing this game of ascension, uh, don't focus on pleasure. Focus on spiritual growth, and then the pain just goes away. So my favorite is to find a super good yoga studio with super good spirit. I tried all yoga studios around. I found one which is exceptional,
Okay.
and just stick to it like once a day. And when I'm sick, twice a day. Go there and just be. You don't have to do anything. Just be in a group of people, and just being in a group of people in a ceremony, it connects you to the healing energy. So that's what I would say.
Okay. Next question is from Psionic Man still. Are triple or quadruple helixes possible?
Yeah, that's my favorite question. Yes and no. I think from what I learned from the aliens and channelings, we ask them that question very often. Most of these next strands are in the ethereal field. They are beyond our physical.
Okay.
So it would be helix in multi-dimensional space with multiple extra strands. In our physical, there is two strands. You can fit one more strand, which I think is once in a while it appears there. It usually is RNA. So two strands of DNA and one strand of RNA, they kind of form a triple helix. Quite often, I would say. There are multiple weird structures which exist, but not along the chromosome, but in small knots in the chromosome.
Okay.
So some special junctions, but I don't think we get DNA triple or quadruple helixes for the long stretches.
Yes, it's just a short
Yeah,
add-ons.
I'm pretty sure that extra strands are in other dimensions. I'm
Okay.
pretty sure there is lots of messages from different sources, including contactees, that there are other dimensional strands. So maybe they are also mutating our other dimensional strands, but we can only see our two strands. So that's what will be analyzing.
Okay. Another one from Psionic Man. Is it possible that human luminosity is a byproduct of said genetic and cellular resonance?
Yes,
Okay.
obvious.
Yeah. All right. Here, last question for him, but he has another one after this. What is Dr. Max's definition of life?
Right. Super cool. Read my chapter. xg1.org. Right after that, I will create a publication section. I will put the chapter there. I temporarily reorganize the site, so it will be there. So xg1.org, it's called WAME chapter and FREYR chapter, two chapters, most recent, 2025. One is accepted for publication already in the book. So water is key, for sure. What is alive? But what DNA is doing, I think, is giving a structure to that water. So there is a code which makes it way more structured on macro level. And DNA works as a portal, a doorway between macro world and quantum world.
Okay.
So DNA-based life is using that portal. And also there is in us, there is a brain which gives us the experience of being dragged forward, pulled forward in time.
Okay.
I don't think many of the life forms which don't have brains, maybe they exist not as focused on current moment as we are. They are like all over their lifespan, like plants. They don't have to be right here thinking about that moment. They can be everywhere. But we are in the brain, there are structures that are responsible for our focus on this specific time. And there are structures which inhibit our memory about past lives, about the future, that is specifically inhibited. So in our illusionary reality, human consciousness, humankind is specially designed to have as little knowledge about psychic stuff. That's what makes us special. We are super non-psychic. It's specially inhibited. So, but when we awaken, we will be much more knowledgeable about past and future and other lifetimes.
Okay. Another question. Could different planetary Schumann resonances, different worlds, naturally from DNA, but the sequencing and folding is different, thus giving us rise to variations and different alien forms on these planets?
I need the help of artificial intelligence to decipher that question. I think I understand the words, but it's not very logical. So let me give one interpretation. So
if the resonance is different, say I'm on a different planet, is that
on a different planet?
Yeah. Say we're not on. It says right here, could a different planetary Schumann resonance affect the DNA and the sequencing?
So you say a human is transported to another planet. Would it mutate to adapt to another planet?
Yes.
I know there are humans in Pleiades star called Taygeta, the planet called Terra. There are genetically humans. And they just live fine with that. So that planet is very close to Earth by qualities. So you can remain normal human like that there.
Okay.
But the aliens which come here, most of them are only temporary here, and they have to wear energetic body suits like space suits to be here. And if they are without suits, they are damaged by everything, air, vibrations and stuff. So that's why they make hybrids. So they can actually use their spiritual design, their souls already with incarnations, to come here and be as completely here as possible. Okay. So they need to modify us to be compatible with the planet, but also with them. So that's why they do a lot of hybridization.
Okay.
So they do that artificially most of the time. And if we move there, we probably will want to modify ourselves to match another planet. But on the ships, humans are pretty fine. So many humans live out there in the ships and artificial bases. So it can be artificially, our vibrational planet vibration can be artificially reproduced elsewhere. It's not that hard.
So we've got last question here. Is just certain people or all humans? If it's certain people, why are they chosen to be hybrids? Is it down the family? We're doing family lines, or are we choosing certain people?
So how do aliens pick and choose hybrids? Great question. I should mention. So in 2009, I started researching that. I started looking for channelers. In 2013, I was given a channeler. In the spring, late spring, early June 2014, I was given a channeler, Jim Charles. And as soon as I started speaking to the aliens, I realized how little they know about us. And I developed a plan that we should volunteer to be hybrids. And we should volunteer to visit, so we don't have to be secretly abducted. We will volunteer to be abducted, volunteer to become hybrids, and help to save humanity, to transform it, to actually make it telepathic and get out of the trap where we are now.
Yeah.
Out of the crisis. So I wrote a book back then. It's called Welcome to Earth. I spoke about how humans became what we are, and deceptive and confused and non-telepathic, and how we use the voluntary visits and voluntary hybridization to be there. So lots of people volunteer on the soul level. Some volunteer on the physical level. Some of my group, we asked to be hybridized for hybrid children. And lots of my friends have hybrid children up there. It's hard to prove, but we believe that. We visit them spiritually there, and we have some connection to them. So it's not a lot of fun to be an autistic child and to be an autist and to be a hybrid here. So I guess the most advanced ones, most brave ones, or most crazy ones are the ones who volunteered for that. And also just some of the military, they were just there. So some maybe didn't volunteer. Maybe on the soul level they agreed, but some just happened to be there. So bloodlines, yes. One of my branches goes into rabbinic bloodline, connecting me to Maharal of Prague. So many of them were studied Kabbalah, and at least that line is always have been strange. So we are connected to angels, I guess, angels and spirits.
Yeah. Well, sir, Dr. Max, I've had a wonderful time on this discussion. Wonderful questions from our audience members, and we can connect. Actually, we'll connect after this because we actually have a Galactic Family podcast Discord channel. We have a lot of the guests that actually came on the podcast on there as well. So if you want to collaborate or try to maybe get more people to know your information, you're more than welcome to join.
Yes, absolutely. Okay. Yeah, I want everybody to participate. That's a toy which can be shared. Let's do it all together.
Yes, exactly, sir. Exactly. So, sir, wonderful to have you here, and I will talk to you after this then, sir.
Absolutely. Thank you very much for having me.
Yes, sir. Have a wonderful day, sir.
You too, sir.
Oh, it's that military in my head again. Dr. Max, Dr. Max, Dr. Max. I got to get that in my head. But wonderful guest to be on the podcast. I can't wait for the next one. We have another wonderful guest that will be coming up on the podcast. It will be on Quantum Pass and Raelian Science, the vision of Natalia Maurice. So I've got her coming on, and she will be here. Let me get my calendar again here. So we've got her coming on. There we go. Quantum Pass and Raelian, November 7th. And that will be at 6:30 p.m., November 7th. And we also have our wonderful contribution page that just happened. We did it, guys. We did it. We got the funds. We were able to go through, and we're going to have a live celebration on November 12th at 11 o'clock at Danville Area SPCA Center for the animals. We have beds. We have we have beds, food, treats, all sorts of different goodies for the animals there, so they can, so that we don't forget them in this time of need. All the shelters around the area, they really need your help. It's December. They need blankets. They need beds, food, anything that you can give them. It'll help their furry animal and help them find their forever home. That's what we need to do here, and that's what we're starting, the Animal Rescue Outreach Project. So let's keep it going, and we can make a difference in just one furry friend, one person at a time. And that's what I'm happy here. I'm happy that we're doing it. We got it. And we're making a difference, even though it's small. It's small now, but we're going to make a bigger difference in the future. And I know we will. So we also have something that I've been looking at, which is, let me bring up the actual, there it is. Welcome to Phenomenon Pulse on the Galactic Family podcast. We will be doing a weekly UFO strange news show weekly, starting probably after November 12th. I'm getting everything around now. It's going to be called Phenomenon Pulse. I love the name. And we'll be getting it on like everybody else. We need to figure out what's going on out there in the universe. And let's get this going, guys, Galactic Family. And I'm just happy to see you guys all here. Happy you guys were here for the wonderful guests we had. And let's get ready for the next one. Quantum Pass and Raelian Science. This can be a great one. Hopefully I'll see you guys there. Have a wonderful day, evening, night, wherever you guys are in the world. And as always, I love you guys. I'm not going anywhere. The Galactic Family is here to stay, and we have one final thing that that I wanted to show, or actually to play, which would be, we have a special song that was produced by yours truly for Jeremy Corbell, to get him into the topic and into the discussion here on the channel. Let's hear this out.
They said the stars were silent, but you saw through the dark. You lit the fire of questions, and now you've left a mark. Voices rising higher, the truth is breaking free. Jeremy, keep pushing for all the world to see. No fear, no chains, we're stronger every day. The sky won't hide, the truth will find its way. Keep the light on, don't let go. Jeremy Corbell, the world must know. Breaking silence, chasing skies, UFO truth before our eyes. Keep the light on, lead us through. The fight for tomorrow belongs to ya. The walls are slowly falling, the crowd begins to cheer. The world is finally listening, the moment's drawing near. Through shadows and through silence, you never lost a fight. Now every heart is burning for answers in the night. No fear, no chains, we're stronger every day. The sky won't hide, the truth will find its way. Keep the light on, don't let go. Jeremy Corbell, the world must know. Breaking silence, chasing skies, UFO truth before our eyes. Keep the light on, lead us through. The fight for tomorrow belongs to you. Oh, we'll rise together. Oh, the truth forever. Oh, no turning back, the stars are ours tonight. Don't let go. Jeremy Corbell, the world must know. Breaking silence, chasing skies, UFO truth before our eyes. Keep the light on, lead us through. This fight for tomorrow belongs to you. Yeah.
All right, so that's the new song on the Galactic Family podcast, and there's the ending. All right, guys. So that was the new song by the Galactic Family podcast. Keep the Lights On, Jeremy Corbell. Let's get him over to our channel and see what we can't do and pick his brain on Phenomenon Pulse. All right, guys. Have a great one.